Okay, maybe we'll go ahead and get started. So thank you very much, everyone that's participating for joining us. I'm Robert Thorne, and I'm here with my colleague Meredith Calvert. We're both scientists at Denali Therapeutics.
Great, yeah, and thanks everybody for joining us today. So what we're going to do is in a minute or so here, we're going to switch over to the video of some content we've recorded. So during that video, you get to watch Robert teach us all about the anatomy of the brain, and we're going to learn about some of the really important structures in the brain. And then you're going to join me to take a look on a couple of different microscopes at how we use the microscopes to look inside the brain and inside the cells of the brain. So we hope you enjoy that, and we also hope that you stick around because at the end we're going to have a Q&A session happening live here with Robert and myself. So as you're watching the videos, if you think of any questions, anything at all that you'd like to ask us, we invite you to do that at the end of today's session. And we're looking forward to hearing from all of you. We had some really great questions during the session yesterday. So looking forward to more of that. So, Robert, if you want to cue us up there for the video.
Yes, I will cue up the video.
Hi everyone, welcome to Denali's virtual booth at the Bay Area Science Festival. We are a company that is situated in South San Francisco and dedicated to developing therapies against a very devastating set of disorders known as neurodegenerative diseases. This includes Alzheimer's disease, Parkinson's, and ALS, among others. So today we have two exciting sessions in store for you. The first will take you into our labs for a virtual tour. You'll also meet a fantastic set of scientists and MDs that are driving a lot of the research and clinical work done at our biotech company. Our second session will give you a macroscopic and microscopic view of the human and rodent brain. We'll see how a human and rodent brain compare against one another and how mouse models can be used to understand these different diseases. And then finally, we'll take a dive into hands-on home activities. So let's begin.
Hi, everyone. So after watching our demonstrations from Dr. Robert Thorne and Dr. Meredith Calvert, I hope you're ready to dive in to do some neuroscience at home. So today we have two activities for you, which you will find in our digital content. The first is a build your own neuron at home. So in my demonstration, I have pipe cleaners, but you can use anything you find at home, like recycled materials, such as straws, popsicle sticks, or even candy. So if you click on the worksheet, you'll find some information about what a neuron looks like. So neurons are the building blocks of the brain. And you'll find some instructions on how to build all the different parts of the neuron. So using pipe cleaners, you'll make little circles that represent the nucleus or the brain of the cell and other parts of the neuron. Our second activity is a build your own brain hat. So you'll also find templates that look like this, which you can print out either a larger size or just a letter size. And this will allow you to build your own brain at home. So here you'll be able to cut out this template and fold it and glue it into the shape of a hat that you can wear. And on one side of the brain, you'll see all the different functions of different parts of our brain. This section helps us learn and remember, and this section helps us for seeing. And on the other half of the brain, you can read about what goes wrong when different parts of the brain are affected by diseases. So we hope you'll enjoy these neuroscience at home activities.
Robert Thorne.
And I'm Meredith Calvert, and we're scientists here at Denali Therapeutics.
I'm a neuroscientist, which means that I study the brain, the central nervous system, and diseases that affect those tissues. I'm also an adjunct associate professor at the University of Minnesota. I've previously been on the faculty at 2 institutions. I was a professor at the University of Wisconsin in Madison, and I was an instructor at New York University School of Medicine in Manhattan.
And I'm an imaging scientist. That means that I use microscopes to look inside cells and tissues and other biological samples. And I also use computers to analyze those images and extract biological data from them. Before joining Denali a year and a half ago, I was the director of the Histology and Microscopy Core at Gladstone Institutes at UCSF, where I looked at hundreds of different types of biological samples from many different scientists. Since joining Denali, I use my microscopes to look specifically inside the brain and try to understand what's happening inside the brain in some of these types of diseases.
So what do we do at Denali Therapeutics? We basically research and try to develop new therapies for neurodegenerative diseases. So we're a neuroscience company. We focus on disorders such as Alzheimer's disease, Parkinson's disease, amyotrophic lateral sclerosis or Lou Gehrig's disease, and other disorders.
And today, Robert and I are going to take you on a tour inside the brain. You're going to start off in the lab with Robert, where he's going to show you some human brains and also some brains from other animal species that we work with here. He's going to show you some of the most important anatomical structures when we're thinking about diseases of the brain. And when you finish up in the lab, you're going to join me in the microscopy room, and we're going to use a couple of different types of microscopes to go deep inside the brain and actually look to see what's happening inside those cells and structures at much higher magnification.
So you ready to get started? Let's go to the lab. Okay, so here we are in the lab, and I'm going to take you through a sort of tutorial on the brain and compare across species. So I've been doing neuroscience for about 30 years. And for most of that time, I've been researching how to deliver proteins like antibodies and other large molecules into the brain. Now, a lot of this work is done using mice and rats as experimental subjects. I get asked a lot about how much studies, experiments in rats and mice and rodents really can translate to learning things that could be useful to treat diseases in human beings. And it's a reasonable question. Rats and mice are a lot smaller than we are, and they're going to have brains that are a lot smaller than ours. And so oftentimes, we might use a model like this to talk about and sort of go through the different parts of the brain. So I'm going to tell you a little bit about the anatomy of the central nervous system. But I actually have some real specimens over here. Most of the experiments that are done in neuroscience, particularly at a company like Denali, are done in mice because they are more genetically tractable. There are transgenic models of diseases. There are more disease models that are available. So you just saw the model here, and now we have an actual human brain. So this is from an old person that passed away and left their body to science. And so brains like this are sometimes used for teaching purposes. And this is a brain that's been in fixative for a long time and is used for teaching. I've also got a half brain, a half human brain that corresponds to this half, the right side. And we'll look at that in a little bit. And last, we have a couple of rat brains, as well as a couple of mouse brains. So the first thing that you should notice when you look at these different brains is the different size. So the human brain here, and then the 2 rat brains, and the 2 mouse brains. So the rat brain is about 5 times as large based on mass than the mouse brain. And the human brain is over 3,000 times as large as the mouse brain based on mass. And so let's quickly talk through some of the different structures in the human brain. First thing to notice, so we'll sort of go from below up to higher up. this is part of the spinal cord right here. And it's been cut. Spinal cord would go much longer if we had the whole thing here. And so the spinal cord is concerned with the limbs and the trunk of the body. So there are motor regions in the spinal cord that are concerned with movement of the limbs and the trunk. And then there are regions that are devoted to sensation. So pain, touch, position sense of the limbs. Moving up, we finally get up to the brainstem. So the brainstem consists of three parts: the medulla, the pons, and then the midbrain, which is a little bit hard to see. And so the brainstem, the spinal cord has nerves that come out, and they're the spinal nerves. The brainstem has nerves as well that are called cranial nerves. So there are 12 cranial nerves for the brain and 31 in the spinal cord. And one of the things to point out is there are the nerves. There are some nerves. They look kind of like little pieces of linguine or spaghetti. And each of these nerves has a very specific function. This nerve goes to 4 of the 6 muscles of the eye. This nerve is primarily sensation to the face, the trigeminal nerve. And then there's another nerve over here, this one, which is the vestibulocochlear nerve and mediates the sense of hearing and the balance from the semicircular canals. One thing you also notice is that there's this structure here sitting on top of the brain. That's actually the blood vessels. So the blood supply of the brain is quite beautiful and intricate. And all of these different parts have specific names. These are the vertebral arteries coming together to be the basilar artery. And there's a nice circle here in the middle. So like a circular ring of blood vessels. And that's named after a 17th century physician, Sir Thomas Willis. It's called the Circle of Willis. So lots of things happen in the brainstem. It's complicated. Arousal, respiration, breathing, things like that. In back of the brainstem is this big piece here, and that's the cerebellum. Cerebellum is focused on coordination and balance. Very important structure. So as we move up, we can see then the cerebral hemispheres. Above the brainstem, there's some structures inside, but we'll skip those for now. So the cerebral hemispheres, we've got the left side of the brain and then the right side of the brain. They exhibit what's called bilateral symmetry. So all of the sort of structural features of this side and the landmarks are the same as they over here on this side as well. This particular brain, it looks a little bit different from the left side to the right. And that's because this side has some structures that are still on it called the meninges, specifically the leptomeninges. And you can see blood vessels running in this structure here. It looks a little bit like cellophane. So on this side of the brain, it's been taken off, those leptomeninges, so that you can actually see all the different folds. And you can also see, some of the blood vessels that have been left behind. Those are all blood vessels there. So all these different folds have names, and the valleys in between them also have names. So the folds that stick up are called gyri, singular gyrus, and the spaces in between them are called sulci, singular sulcus. And very quickly, we can use some of these landmarks to define the different lobes of the brain, and I'm going to do that in a moment. But before I do, I just want to remind you that one material that is available for you to download, here we go, is this brain map. And if you want, you can download this and follow the instructions to put it together. You can also do it with an 11 by 17 paper and make a small, so this is meant to be a brain hat. You could put it on your head. If you do it with an 8.5 by 11 paper, it'll be small, but it also could be just a little model of the brain. And so when you finish doing that, it'll look like this. And that's something we can talk about as well in the Q&A. Okay, so lobes of the brain. There's some major landmarks to be concerned with. The first one is the lateral sulcus, which is right here, below which is the temporal lobe. The other major landmark is a sulcus that runs like this. and it's the central sulcus, which is right here. The central sulcus separates the frontal lobe from the parietal lobe. And then the last of the four lobes is back here, which is the occipital lobe. So the frontal lobe has lots of different parts, but suffice it to say that some of the major functions for the frontal lobe are the primary motor cortex and the precentral gyrus, which contains motor neurons that control movement for the body. There are also some other motor areas, areas that control the frontal eye field, so saccadic movement of the eyes. And then finally, this prefrontal cortex area, which is important for executive function, personality, and tragically is often affected in some of the diseases that cause dementia, like frontal lobe dementia. The lobe that is just behind the central sulcus is the parietal lobe, and in that lobe we have the postcentral gyrus, which contains the primary somatosensory cortex. So that gyrus contains sensory neurons that are important for conveying basically the awareness of touch, pain, temperature, where your limbs are in space, where your head is in space. There's also a structure that's important for the awareness of the body, self-awareness. Moving around here, again, central sulcus, the lateral sulcus, which is right here. Below it is the temporal lobe. It's a very important structure here, sort of peeking out of that lateral sulcus, which is the transverse temporal gyrus or the primary auditory cortex. If we move around the temporal lobe and we get to the medial portion, the portion that's most in the midline here, right here, this is the parahippocampal gyrus, and there's a little bump next to it called the uncus. These two structures respectively contain the hippocampus, which is important for learning and memory, and then the amygdala, which is important for emotional content. And tragically, these are often affected very early on in Alzheimer's disease, medial temporal lobe. The last lobe we have is the occipital lobe, which contains the primary visual cortex. So this is where vision happens back here. If we look real quickly here in this section of the brain, it's a horizontal section, we can see the fluid-filled ventricles. These are the lateral ventricles of the brain. And so these are fluid compartments. And this is the choroid plexus that secretes the cerebrospinal fluid. So one thing that is important to emphasize is that all of the parts that are in the human brain are basically also in the rodent brains, in the rats and the mouse. And that's super important, because what that means is that we can do experiments and we can use models of disease in these animals to learn things that are very important for developing therapies for disorders like Alzheimer's and Parkinson's and what have you. There are some differences, though, that are interesting to point out. And one is, we just finished looking at the hemisphere here, and I was pointing out all the folds and the valleys. And I want you to look at the rodent brains, the rat brains here. The hemispheres are here, here, and then the mouse brain, hemispheres are here and here. And the thing you should notice is that the rodent brains don't have those folds. The reason for that is that the human brain uses these folds to pack more neurons into a tight volume. And indeed, actually, this is one of the things that makes our brains unique. The cortex, which is this area out here, the outermost part of the hemisphere, our cortex contains the most neurons of any species on the planet. That is super important. Looking at folds, another thing I would point out is that the structure of the cerebellum that I mentioned earlier, that's important for coordination and balance. If you look at it closely, it's super folded, much more folded even than the hemispheres. And that is because 80% of all the neurons in the brain are in the cerebellum. And if you look closely at the rodent brains, you look at the cerebellum there of the rat, you'll also notice that that structure is folded as well. So the hemispheres are not folded in the rodent, but the cerebellum is. Nature's way of packing more into a tight space, tight volume, is to fold. And so it turns out that in the rodent, as in the human, 80% of all the neurons in the whole brain are in that structure, the cerebellum. Another interesting comparison between the rodent and the human that's actually in favor of the rodent is the olfactory bulbs. The olfactory structures, the olfactory bulb is here and here in the human. And then you see them down here, these structures in the front of the brain, in the rat, and then in the mouse. And the thing you should notice is that relative to the other, the rest of the brain, the rodents have way bigger olfactory bulbs than we do. In fact, the rat brain olfactory bulb is almost similar in size to the human. And the reason for this is somewhat obvious. Rodents use their sense of smell much more than we do. Olfactory bulbs have actually regressed in primates, like human beings. And so this is something where the rodent actually is far superior. We use our visual sense much more than the rodent does, and we rely less on our sense of smell. Now, I'm often asked if brain size is simply one explanation for what makes human beings so smart, for instance. And it turns out that's not really the case. You know, it's not true that our brain is the biggest brain of any species on the planet. There are lots of animals that have bigger brains than we do. Elephants have brains that are approximately 4 times as big as ours. But again, the difference between an elephant and us is the number of neurons in the cortex. So 86 billion neurons in the whole human brain. An African elephant has 257 billion neurons. We have the most neurons in the cortex. What are all those other neurons in the elephant? 257 billion versus 86 billion. Well, it turns out that while we have 80% of our neurons in the cerebellum, an African elephant has 98% of their neurons in that structure. Coordination and balance, why would that be? The elephant has an extra limb, essentially, an extra appendage, which is the trunk, and that needs to be coordinated with the rest of the body. So that's one of the explanations. So there are lots of other things we could focus on with this tutorial, but I hope that gives you a sense of the beauty of the brain, the differences across species, and look forward to addressing any other questions you might have during the question and answer period that we'll do with Meredith. Now let's go over and go look at some microscopy demonstrations with Meredith.
So thanks to Robert for giving us that great introduction to the brain's sort of larger structures. So now what we're going to do is we're going to use the microscope to look inside the brain at an even closer detail. We're actually going to go inside the tissue itself. So if you take a look here, what I actually have is very, very thin slices of, in this case, mouse brain that have been mounted on this slide. What we then do is we stain the tissue with different types of dyes that allow us to see some of the different structures that are within the tissue. So it's almost like looking through a stained glass window. We're actually going to use light, in this case just like a regular lamp light, that's going to shine through this tissue and give us a view of what's going on inside there by looking at the stained tissue. So we're going to go ahead and put our slide on here, and let's take a look. So the adult brain is made up of around 100 billion cells, and through the connections, or known as synapses, each cell can connect to around 10,000 other cells. Most of the cells in the brain are known as neurons. Of course, these cells are too small to see with the naked eye, and that's why we're using a microscope to let us see these very small cells. So as you can see here, we can actually see the individual neurons and some of those connections that are within them. If we think about it, all of the information our body needs to survive, for instance, that you're hungry, that you've stubbed your toe, remembering your mom's birthday, all of this information travels along these neuronal connections, like you can see here. In certain types of disease, such as Alzheimer's disease, the neurons of the brain begin to die, and these connections break down. At Denali, we're trying to understand what goes wrong in the brain in these diseases so that we can find medicines to help prevent or repair the problem. One thing we know about Alzheimer's disease
is that abnormal amounts of certain proteins, such as amyloid proteins, accumulate in the brain and form structures known as plaques, which are large clumps or tangles of proteins that form in between and around the neurons and contribute to neuronal cell death. What we're looking at here is a healthy brain, but now let's come over and take a look at a diseased brain. You see these large dark clumps in and around the neurons? So these are those amyloid plaques we were talking about. And this is really what's causing the problem within this particular type of disease. Okay, so this is another type of microscope that we use for studying our samples. And this is actually what's known as a confocal microscope. With a confocal microscope, we're no longer using normal visible light or normal lamp light. Instead, we're actually using lasers to illuminate our sample. And the benefit here is we can stain our sample with different types of fluorescent dyes, such that the different structures are stained in different colors. We can then use different color laser light to illuminate these samples. In this case, the fluorescent or the laser light hits the sample, and the sample actually reflects that light back into the instrument where we can then record it. And again, we can use different colors of light to show us some of these different structures. A confocal microscope allows us to go deeper into the tissue and also to see things at a much higher magnification than is possible with a normal microscope. The other thing that we can do with a confocal microscope is three-dimensional imaging. So what we actually do to do this is we capture an image at a particular focal plane within the tissue. We can then adjust the focal plane, moving deeper into the tissue, take a second image, move deeper, take another image, and this ends up leaving us with a stack of images of our sample. So you can see here, we're actually moving through the tissue one slice at a time, but these are optical slices that we've taken through the tissue. The cool thing here is that now we have a whole stack of these images. We can now project them into 3D. So let's take a look at that here. Let's see. So now here's a whole stack of images. And you can see we actually can recreate the three-dimensional structure from within the brain. And this allows us to see things at obviously a much higher level, but also we can actually see what's going on inside the cells and inside the vessels that make up the brain. In this video, we see a mouse brain stained with one marker that shows every cell in the brain marked in magenta. Notice how these two areas, the cerebellum and the olfactory bulb that we learned about from Robert, notice how these areas are so much brighter due to the really high density of cells in these parts of the brain. The turquoise marker is showing us another protein which accumulates in the damaged areas of the brain in the Alzheimer's disease patients. Here's another movie we made using confocal microscopy. In this case, we've marked the cells in turquoise and the vessels of the brain are marked in red. We're using three-dimensional rendering to be able to see the structure of the vessels and the cells, and even see some of the proteins that accumulate inside the cells, shown here in yellow. As a microscopist and a biologist, my job is to make images and movies that help us to better understand what's happening inside these cells of the brain. One thing I love about my job is that I get to create beautiful images that not only illustrate the amazingly complex biology of the brain, but also provide important information that can help us better understand human disease.
So thanks for being with us. We hope you enjoyed the virtual tour of the brain and then microscopy of the brain. To show you forward to having you with us at a question and answer that's part of this session. So any of you that have been on this with us.
Festival where you can learn a little bit more about the different types of people working at Denali, other than just a scientist, and what it takes to put together a biopharma drug discovery company. So I look forward to seeing you there too, and we hope you enjoyed your time with us today.
Thanks for joining us.
Thank you.
All right then, so thanks very much. I'll just switch back to our PowerPoint. And we will now commence the question and answer part. For those of you that are participating, Meredith and I would be happy to address any questions you might have while you're thinking of questions. So this Bell to Brain Hat, this template that was mentioned in the introduction, is a resource that you can find on the Bay Area Science Festival website and download it. This basically this whole thing. And on there are some questions. So if it takes a little bit of time, we have a question. Maybe I'll let Meredith, should I take that or should you?
Sure, well, let me take a look here. So the question is the loss of vision and hearing with age considered a degenerative brain condition? Yeah, that's a great question. Robert, I'm gonna let you answer that one.
Yeah, so when we did this yesterday, we were asked about, what causes these brain diseases that we study. And one of the things that I said is that, we don't fully understand what causes Alzheimer's disease, Parkinson's disease. Some other diseases, there's a more prominent genetic component. But what I would say about, so loss of vision and hearing, what we do know is there are risk factors for these diseases, and the biggest risk factor for a number of these diseases is age, advanced age. And so when I think of your question, loss of vision, I think of an unfortunate disease that affects people's central vision, which is called macular degeneration. And macular degeneration, Alzheimer's disease, Parkinson's disease, all of these conditions are what are called age-related. And so the older you get, the more likely you are to suffer from them. And hearing is the same kind of thing that, typically people have deficits in their in the auditory system as they, it's more likely as you get older. But you can definitely focus and point to specific diseases that are associated with age. And just to flesh out the answer, so Alzheimer's disease is a really good example of this, where you can say about 5% of people 65 and over have Alzheimer's disease. If you consider only individuals age 80 and over, the number jumps up to about 30 to 40%. So the older you are, unfortunately, the more risk, the more likely you're going to have these conditions. And this same thing applies to age-related macular degeneration, a disease of vision.
That's great. All right, we've got another question here. We've been getting a lot of great COVID questions, of course, recently, which so we'll do our best to address those. So the next one is, what is brain fog and its causes, and how is this related to long hauler COVID-19 patients? So Robert, I don't know if you have any thoughts on that one.
Yeah, so COVID-19, the virus that causes COVID-19, when it gets into the body, it goes to different organs and then produces the effects. There's a lot of inflammation that underlies what you have to deal with COVID-19 and treat it. And so, we're all familiar with the inflammation that happens in the lungs and the respiratory system that often puts people in the hospital. They're unable to breathe normally and get oxygen into the body. There are a lot of neurological symptoms associated with COVID-19 as well, though. And probably the biggest one I'd point out is deficits in the sense of smell and taste, which most COVID-19 patients that are symptomatic have something like this. And indeed, it's among some of the first symptoms that people notice. So I did talk about in my video about the sense of smell, and just maybe remind you about that there's a particular structure in the brain, the olfactory bulbs, and the olfactory system that's devoted to that. So why might COVID patients have deficits in the sense of smell? It probably has to do with the virus actually gaining entry to the brain and propagating to some degree within the system, within the olfactory areas of the nervous system, and creating a deficit that's associated with what that structure does. And so the olfactory bulbs actually are pretty, that's not uncommon for them. So there are a lot of viruses that actually gain entry to the brain through our nasal passages. You inhale air, and very high up in the nasal passages is the olfactory region. So brain fog it's not really a technical scientific term, but so if you have viral propagation, so the virus gets into the brain and then multiplies and triggers inflammation broadly in the brain, particularly in that frontal lobe area. The frontal lobe area, as I mentioned, is where you have this sort of executive function personality. If you had a lot of virus in that area, then you might expect, or even the temporal lobe, the medial temporal lobe, like I mentioned, for learning and memory. So same sort of thing as with the olfactory system. If you have a lot of high titer of virus in those areas, and then that was associated with some inflammation, then what those areas of the brain do starts to be affected. And so that's a short answer to a complicated question.
Very complicated question. Yeah, as with many things in the brain, there's a lot more that we probably don't know than what we actually do know. And of course, that's even more true in the case of COVID, where we really are learning more about this disease every day. So the next question we have, and perhaps I can try and answer some of this. So we have, what are some common subspecialties of neuroscientific research? So myself, I'm actually relatively new to neuroscience. I had certainly worked with neuroscientists in the past, but I really came into this field by way of imaging science, because that's really my specialty. But that's just kind of one example of how you can be a specialist in neurobiology. But within that, there's behavioral neuroscience, there's psychiatric neuroscience, looking at some of the more psychiatric disorders. Obviously, neurophysiology, or just kind of the metabolic regulation of the brain and associated structures is a whole other area of the field. Gosh, lots more. Robert, what are some of the other ones I'm not thinking?
Yeah, I mean, that's a good recitation of the different disciplines in neuroscience. I think another question we get, maybe I would ask you to address, Meredith, is, at a company like ours, we're a neuroscience company, we're a company that focuses on neurodegenerative diseases and treatments. We have a lot of people here that are not neuroscientists, like all the different sorts of people that work at a biotech company like ours that's focused on diseases of the brain. Maybe you could speak to that a little bit too, Meredith.
Sure. Yeah, absolutely. Yeah, it's, I mean, so we have, again, we have a panel discussion actually as well. And there's a video associated with that, which will give you the opportunity to meet some of the other folks working at Denali, who are perhaps not, traditional neuroscientists, but, of course, we also have, medical doctors. There's people whose specialties are dealing with clinical research and setting up clinical trials. We have people whose specialty it is really to interact with the FDA, the Food and Drug Administration, which is who oversees most of these clinical trials and helps to decide which drugs can move through into the clinic. So of course there's safety monitors and pathologists and toxicologists. Yeah, really runs the gamut. I would say if you're interested in neuroscience, but perhaps, coming in from another area of specialty, or, aren't really sure exactly what direction to go in, what I will say is that, again, at a company like Denali, we have people from all backgrounds, but the wonderful thing is we actually all get to learn about neuroscience from within the company, because of course everyone's kind of sharing their research, and we're learning from each other as we go. So, I know for myself, I've certainly learned more neurobiology in the past year than I had ever known previously. So yeah, takes all kinds of backgrounds to have sort of a successful research program. So I'm not seeing any more open questions there right now, but one of the things I thought perhaps we could briefly touch on that I think we didn't go into was the complexity of the cell types within the brain too. I mean, we sort of talked about neurons generally, but that's also not the only type of cell within the brain. Of course, we also have the vasculature, which has endothelial cells, which is keeping the blood flow to the brain. There are specialized types of immune cells within the brain as well. And those actually play a fairly large role in a lot of these neurodegenerative conditions too. So even, beyond just the complexities of the structures of the neurons, there's the complexities of the different cell types as well. So yeah, many layers there.
So we have another question with the gut-brain axis. Could targeting bacteria in the gut be a solution against Alzheimer's disease? And I'm going to expand the question, or other neurodegenerative disease conditions. And So there's a lot of research actually that has been focused over the last decade or so on this sort of gut-brain axis, the communication between the gut and the brain. There is a nervous system that's part of the gut. It's called the enteric nervous system. And we know a lot more about so what's sometimes termed the microbiome, essentially the bacterial content, all of the sort of complexity of the different organisms that are in the GI tract and how they affect the cells of the gut. And then those cells, and sometimes even the bacteria, the microbiome, can release factors that then would get into the body and travel in the circulation and reach the brain. And so it is known that the microbiome of the gut can affect, for example, the blood-brain barrier, this feature of the brain's blood vessels, whereby they're very tight and they restrict the free passage of a whole host of different things that are circulating in the bloodstream. And that blood-brain barrier is actually one of the things that we study very carefully at Denali, because in order to deliver therapies into the brain, you have to get across that blood-brain barrier in some way. And Meredith had some beautiful slides where you could actually see those blood vessels in the sections that she was showing. So it is very likely, getting back to this sort of opening question, the theme of that we don't fully understand what causes these diseases and all the factors that influence their progression, that can make them worse as well as make them better. And certainly diet, again, the microbiome, sort of the detailed composition of your gut outside of the body is one factor that certainly plays a role, and we're learning more about that. Parkinson's disease is a disease actually where there's a lot more pathology that happens in the gut that's fairly well established and well known, and the gut is actually one of the earliest places where you see the pathology, some of the things that are happening that are abnormal in Parkinson's disease. And so if you test Parkinson's patients very early on carefully, you often find that they have symptoms that are related to that you might imagine that would be associated with some sort of dysfunction in the gut. So there's a lot of research that's done in that area. Most of it is in the academic realm at universities. So we at Denali don't specifically study that.
Yeah, and just to kind of expand on that a little bit, another thing that we do know is that there's a big link between kind of diet and inflammatory states in general. So, there's sort of an inflammatory cycle that can be kicked off in the body. And we do know that high fat, high sugar diets and so on can contribute to increased inflammatory states. And there's certainly probably a role for that in I would say, most human diseases, but certainly that includes diseases of the brain as well. And so then similarly, there has also been evidence that sort of diet modifications, increasing exercise, these kinds of things can change the progression of these diseases as well after diagnosis. So there's certainly a link there between sort of general health, which obviously diet is a huge component of that too. So, but again, another area that we're really just now beginning to understand. So,
yeah, I might. So on this brain map, Belda brain hat, I have some questions, and we kind of got into a little bit in the video. I mentioned that the human brain is very folded. So this is something that's called gyrification. And so all of these folds, and then we talked about the rodent brains, and I'll show you here the rat brain not being folded. So it's much more smooth. And the reason for that is this folding allows for a lot more neurons to be packed into that area. And so back to this question, what makes human beings so smart? I said in the video, it's not that we don't have the largest brains in the animal kingdom. That's not the answer. It's also not the answer that our brains have more neurons than other animals. I think I also mentioned that elephants have a lot more neurons than we do. But the answer is more neurons in the cerebral cortex, in this outermost layer. And so I also have this model, and my model, kind of like the brain I actually had, I can open it up. And so if we open it up, we can actually see what's referred to as the white matter, and then this outer bit, which has a little bit more coloration, which is called the gray matter. And so it's the gray matter that we're talking about, this outermost gray matter, which is the cortex. And so that's the part of the brain where human beings, we have 16 billion neurons in that cortical gray matter. Whereas to compare, an African elephant only has about 5 to 6 billion neurons in that outermost part. The other thing that I mentioned was that the folding, if you look at the cerebellum here, you can see how the cerebellum's really folded, much more folded than the cortex. And I mentioned that 80% of the neurons in our brain are there. So most of the rest of the other 20% are in that cortex, but 80% are in the cerebellum. And if you look again at the rodent brain and the rat, so I can see what I'm pointing to. So over here is the cerebellum, and if you look closely, you can see that the rat cerebellum's also got those folds like ours does. And so the rat cerebellum also contains 80%. 80% of the total neurons in the brain are here in this structure, coordination and balance. And so this is when Meredith's looking at these structures under the microscope, and she mentioned this. You can see this, you can see all these cells. I don't know if you want to say anything more about that, Meredith, but it's unmistakable.
Yeah, absolutely. Yeah, I mean, one of the neat things about, fluorescence microscopy in particular is that the brightness of the light that we see tells us something about the amount of the protein or the amount of particular structure we're looking at. So in the case of that video I showed you, we were just using a general cell marker for cells in the brain. And so you could really see the way that that cerebellum lit up so much brighter than the rest of the brain. It's reflecting just the total number of cells that are there, because again, the intensity of the light increases with the amount of the protein that's there. So Robert, I have a question for you thinking about this. So if we were looking, how does the brain change during human development? So how would a child's brain or a teenager's brain differ from an adult in terms of thinking about this folding and different structures?
Yeah, so the folding gets more prominent over development. So if you look at a brain of a baby, of a fetus when it's in the womb during pregnancy, it's not as folded, so it becomes more folded as you get more and more neurons you get this division, so you're creating a lot of new neurons. And then at birth, the other thing that happens is you get a lot of other cell types, like the glial cells, the non-neurons in the brain, that start to divide and rapidly develop. And so in a human brain, adult brain, you have about 86 billion total neurons and 84 billion total non-neurons or glial cells. And at birth, you have far fewer of the glial cells, so that they start to come on at birth. Cells called astrocytes and cells called oligodendrocytes, which myelinate, essentially insulate the axon of a neuron and allow for the conduction of information. So essentially, neurons, brain cells signal, it's an electrical phenomenon. And when you insulate this sort of this cable that's going to spread the electrical activity and ultimately signal to another cell to fire, to essentially turn on, then when you insulate that cable, the electrical information travels more fast. And so the cells that do the insulation, called oligodendrocytes in the central nervous system, and Schwann cells in the peripheral nervous system, they also come on board around birth, and start to, that's when it starts. And so this is one, if you wonder, like why does a baby have a hard, when they're born, why do they have a hard time keeping their head up? Why does it take them time to learn how to crawl? And part of that is really that this insulation, the myelin that is needed to cover those motor neurons, it takes time to develop, and it's developing over that time frame that you see with babies, where they're quite floppy when they're born, and then eventually they're able to keep their heads up and be upright and crawl and then even walk.
That's very cool. Well, another thing, I love how sort of the structure can really tell us about the function. I mean, this is true all over biology, but, particularly in looking at the brain, we know how that the structure can tell us something about the function there. There was a wonderful study that came out maybe 6 years ago now. And it was actually, one of the nice things with this confocal microscopy, where we can actually do things in 3D, is of course now we can measure volumes. So you're not just looking at two-dimensional things anymore, but we can actually start to look at how shapes change in space, and how the volumes of things change. So in London, the taxi drivers are actually required to memorize the grid of the entire city of London. So, this is sort of pre-GPS, but this is still, in order to get your taxi license in London, you actually have to prove that you have, all of London memorized, all of the streets. And so the study was actually looking at the brains of taxi drivers, and they saw that the hippocampal region, I believe, the volume of the hippocampus inside taxi drivers was actually, up to, I think, 20% larger than in the normal population. And so it's just another example of how they were, taxi drivers' brains over the course of their lives, right, have adapted, have actually changed shape in order to accommodate this additional information that's required for them to do their job. So, well, that's, yeah.
Yes, so the brain is really plastic, and it's developing, changing throughout the lifespan. So there are lots of things that happen when we're born and before we're born, but then throughout our life, then experience what we're exposed to, and the tasks that we have to do are then modifying our circuitry. I have a question for you, Meredith, that, so you showed us a couple of different microscopes in your demonstration, and maybe you could just say a little bit more about like what the different types of microscopes allow you to do. Like, is it one size fits all, that fancy microscope with the screen that you were showing in the microscopy room at Denali? Does that do everything, or do you use different types of microscopes to do different types of tasks, show us different types of information?
Sure. Yeah, that's a great question. So yeah, actually there's many different types of microscopes. And in fact, calling myself a microscopist, I'm a light microscopist, which is quite different than electron microscopy, which is a whole other field in itself. If you want to see things on a really small level, like down to sort of the atomic level, you really need an electron microscope to do that. And the reason is that with light microscopy, we're limited. We can really only measure or observe things down to the size of the wavelength of the light itself. That's the kind of the physical limitation of what we can do. But because of that, the type of light that we use to illuminate the sample, again, dictates the type of information we can get out of it. So that first microscope we looked down, which was more simple, where we were just looking at sort of normal light, or again, it's that kind of stained glass window effect with the tissue. That's what we call a compound microscope. And so with that, we're just, again, using normal light. But also with that, we're imaging all of the light from the structure in 3D. The benefit of the confocal, because by using a laser, we have a specific wavelength, but we also have it only illuminates a subset of the sample. And so we can actually use, by putting a pinhole to block scattered light, we can use that to only image exactly the section that we're looking at. And so we call that optical sectioning, because we're basically using the light to slice the tissue. We're not physically cutting it, but we're only collecting the light from within a single slice. And again, that's really what gives us the ability to do things in 3 dimensions. And there's many other types of microscopes out there. I would say those are my 2 personal favorites. But yeah, there's many different techniques, and it's an exciting field because this is somewhere again where as computers get better and cameras get better and our laser technology gets better, and just the types of things that we can do expands massively, year to year. So it's really exciting. Looks like we have another question that just came in here. So this is Ren saying, do you have any advice for an engineering student whom you've managed to seduce with neuroscience? Is there a place at a company like Denali for an electrical engineer? So yeah, Robert, I don't know if you want to speak to that.
So we actually have a lot of engineers that work at Denali. So we have chemical engineers, probably is the biggest engineering discipline maybe that's represented, and these are the people that help us to make the proteins, the therapies that we develop. If we get to a point where we're going to go into a clinical trial, or we need to make a lot of these proteins, the therapies that we're developing, then it really gets to be a manufacturing question, an engineering problem to scale up and do that appropriately. We also have engineers that are on. So the brain, is this very complex computing organ, and you think of neural networks. So then I can make a connection there to electrical engineering. There is, in understanding some of the data that we're generating, there's even more opportunities for the engineering disciplines. I know Meredith and have talked with some of our colleagues about how we might utilize things like machine learning and artificial intelligence to handle some of the data that is produced. We end up generating, like most, a lot of biomedical research these days, massive amounts of data. And so then it becomes a challenge to, try to pull all the information that is actually available in that we can't. This is a great application of computers, of AI. And I think we're going to see more and more of that in biotech. And certainly I think our company is reflective of that. I don't know if you want to add anything there in our last minute, Meredith?
Sure. Yeah. It looks like we're about out of time. But what I would say is really, anyone with kind of a strong background, I think, electrical engineering certainly takes a certain mindset, certain, problem solving skills, and just it's an analytical way of thinking. And I would say that absolutely that type of expertise would have a place, even if it wasn't hands-on electrical engineering per se. But, I think engineers and scientists, we definitely need each other and rely upon each other heavily. So. And as Robert said, as the technology advances, and also speaking to microscopy, certainly is we really, we need people with technological skills from all types, all backgrounds there.
Yeah, so thanks for joining us. I mean, last thing I might say is that, we're a company of about 300 people, and we very much value and seek to have a diverse workforce environment. And so, for those of you that want to learn more about Denali, you can go to our website. We have a career site. We have a summer internship program. This is really directed at undergraduate and graduate students, and we'll be posting some things on related to that a little bit later this fall. We also have a postdoc program, so that's for people who finish their PhDs. It's something that I head and organize. We have just a great community of people. And so one of the reasons that we're involved in STEM outreach and part of the Bay Area Science Festival is that we really think this is important in neuroscience, in science and STEM, but also in the biotech community. So thanks very much for joining us. And maybe I'll give Meredith the final word.
Sure. Yeah, no, thanks very much. It was a lot of fun, and I hope we've, turned some of you on to neuroscience and, the beauty of the brain, and just what a fascinating field this is. And obviously we have lots of problems still to solve here. So hope you'll think about coming to join us in that. So thanks Great. So much, everybody. Okay.
Thank you.
Bye-bye.